So I'm going to start by assuming that now, after Jonathan's talk, you are very excited about the whole concept of screening, and you really want to, take your favorite biology and build a screen around that. And so in the lab, these technologies that we've been developing are not just for, doing the biology in our lab where we are used to systems biology, but we have been developing these to be easy both for us and for anyone to use, and other labs have been doing the same. So it's really a sort of transformative technology, both in the amount of things that you can do and in how easy they are to do them. And so I'll go really into a little bit more detail on how the mechanics of a screen work, and I'll do that in sort of this first half hour, and then I have also some afternoon time, and then I'll step back again and talk a little bit about the different platforms that people have developed, and then sort of what the exciting things are in the future in terms of screening and multiplexing, and finish up with maybe a few other applications that people have used the same sort of concepts to do really neat tricks and to learn other things besides what gene does what. So again, the progression that we've been thinking about is from classical forage genetics, where you have your pool, usually yeast or Drosophila or whatever. You randomly perturb it, and then you sort of plate all these out. You select for a phenotype, and then you try and figure out what caused that phenotype. And this really is the foundation of most of the biology that we study today. But at the same time, it's really limiting at this point, because, as Jonathan mentioned, you actually have to go and then identify the gene. It's random mutations across the genome. You need to actually go and sort of clone the causative gene. And then, of course, if you're going at random, especially in a haploid genome, you're never going to, in a diploid genome, never really going to reach saturation. You're never going to get every gene tested and see yes or no, does it cause the phenotype that you're interested in. So this is the foundation for then saying, well, what if we had tools to be able to go one after another, say, down the list of 20,000 genes in the human genome, what does this gene do? And so the easiest way to organize that is to put each perturbation in a well and do whatever you want on your defined population with a defined genotype. And so this is same slide as Jonathan, just where the defined perturbation can really be used to test anything. You can do reporters, you can look for growth, you can look for these drug sensitivity things that Jonathan described, how they really, genetics teaches you about drugs, it teaches you about biology. But then, while this works for yeast, it really scales very poorly. You don't want to have a plate hotel in every lab, and especially for human genomes, you'd need like plate super hotels, and there's lots of other issues with it. Like each well has its own microenvironment, and there are a lot of caveats. And so, just with a little math to say, with 20,000 genes, even if you only wanted one siRNA, which is pretty variable. Normally people do at least 4, and just one condition, which doesn't tell you much, and one replicate, you'd 52 plates just to do that screen. And that would be a pretty meaningless screen. So that's where we've really wanted to take the genetics away from and make it a much simpler system. But while recognizing that both these methods had advantages. So classical forage genetics, while it was random, you at least had this one pool. You could evolve it, select for your phenotype, and it was actually really easy to conduct. It was just a lot of legwork at the end. Arrayed screens, you have systematic defined perturbations, so it is very easy to then say, this is the genotype that caused my phenotype. And so these two concepts together is where we have pooled screens, where we have one pool, but using next-gen sequencing technology and using a few other enabling technologies, you can really, in one pool, do the whole genome and do it in a systematic way where you know that each gene is going to be tested. So in a kind of dry text-based view of that, there are really 3 steps in a pooled screen. The first is you have a population of cells and you stably, with a complex, random but defined library, introduce a genotype into each of these cells in your population. And then you evolve the population. Either you treat it with a drug, you just let it grow, you do a FACS-based assay, and your genotype distribution in the population changes. So you can save samples at the beginning, at the end of your experiment, and compare the distributions, and that's where next-gen sequencing comes in. So usually people represent this all as a cartoon, and I think pretty much every screening paper has the same cartoon, where you start with your construction and you put it in cells and they turn colors, but of course in one pool that you actually don't have colors, you just have these barcodes that are in the cells, and then you sequence. And this is a very high-level view. And so in terms of conveying the details of a pooled screen and how you would, in your own lab, want to conduct it, wanted to sort of break down each of these steps and zoom in just one step further. So starting with this, so the library synthesis is not something you really need to do each time. There are lots and lots of libraries on Adgene, and I'll cover the different features of them later. But just to sort of inform this part of the cartoon, it's basically like ordering any other set of oligos. You have a text file, and instead of sending to a company and they send you back 1 tube for each oligo, they print it on these microarray slides. So the same technology is used to print microarrays. So you can make 244,000 on one chip. But even though the cartoon has the chip, we never actually see that. It gets shipped to us in sort of the mainstay of molecular biology, a tube of clear liquid. And it's a little underwhelming when you like make this whole list and then you get a tube like any other oligo. But it makes it very easy to deal with, because then you do some standard molecular cloning. You just need to scale it up a bit to make sure that the library is represented throughout. But you do standard molecular cloning, and you get another tube, and you put it in your freezer, and that's your library. And so it's very, very simple. Just to say what we're doing in the cloning, because that's important in actually what you're putting into your cell line. Most of the vectors that people have used for Cas9 screening are sort of this two transcript system. One is the sgRNA, which needs to be transcribed off a Pol3 promoter. And so basically all of these sequences just have some cloning sites here and here, and they get stuck here. And then everything else is the same. It's a lentiviral backbone. You have your Pol2 promoter, which can produce antibiotic resistance. It can produce colors, so you know how many cells you actually have infected. And sometimes these vectors actually even include Cas9. It's debatable whether that's a good idea or not. One hand, then every cell with an sgRNA has Cas9 automatically, but it's also very big, and it's hard to do all these steps with such a big vector. So that's what a library looks like, is just a very defined constant vector, like you'd use any other vector in transducing cells. But here you have this defined library that you can easily go back and sequence. So the other arm before you actually get into a screen is you need a cell line to screen in. So this cartoon says K562s, but really the hope with these technologies is that in any system you can use the most relevant cell line for the biology you're interested in, or the easiest cell line to screen for the setup that you have. And really these technologies, for the most part, have worked in lots of different cell lines. I think Jonathan had examples of both leukemia and retinal cells in his talk, where the same sgRNAs were ported over. And there are just a few things to think about when you're making a cell line. One thing that I don't have bullet pointed is haploid cells. If you don't really care about the cell line that you're using and you want to do a cutting screen, it still makes it way easier, even with Cas9, to have haploid. And I'll show some data from these screening papers where they compare haploid and diploid, and that's there are certainly things to be said for still working in haploid genetics. There are always questions about whether to do polyclonal or monoclonal to introduce your Cas9 and then sort either a mixed population or down to a single clone. And it really depends on the system, but the key is that every cell in your population at the end needs to have an active, full CRISPR system where all you need to do is add the sgRNA and you get your effect. And the reason is very simple. In the math, if you're counting sgRNAs in a screen, and that's all the sequencing does is count, and all these sgRNAs are in cells with no Cas9, you're counting empty barcodes basically, and your signal is going to be very damp if at all active. So being able to generate nice FACS plots like this, where everything is knocked down or everything is knocked out in the population, is very important as you approach actually doing a screen. So then, how do you do the screen? How do you design an experiment? And the problem is there's no set protocol, because there are a lot of different things that you could do and a lot of things that you have to think about as you're designing it. Yeah.
I had a question about your last slide.
Yeah.
So this one. So
no, the cartoon. Yeah.
Yeah. So what you want is one guide per cell, right?
Yeah.
In the end. So lentiviral particles are heterozygotes. They have two copies of the RNA genome.
Yeah.
So whenever you package them, they probably get two guides per particle. Has anyone in the screening community over the years with SH or even CRISPR now ever single cell cloned individual cells to show that those they get two?
Yeah. So they do package two, but one gets integrated.
So only one.
Yeah. Yeah. And there are definitely things to worry about there. If you have very long, unique barcodes, there can be recombination between the vectors within the lenti, but the single integration is pretty much given. Yeah, thanks. That's a good question. And yeah, actually, I'm sort of omitting this arrow here in my talk, so I'll hand wave it a little bit. So the key is if you're barcoding each cell, you want exactly one barcode per cell, and you need it to be stably in there. And when I say barcode, I mean also the sgRNA. The sgRNA is its own barcode. And so you use lentivirus because it integrates into the genome, and you have to sort of titrate it such that you get just about one sgRNA per cell. And so you usually infect to about 30 to 50% of your cells get the infection, because if you go higher than that, then some fraction of the cells will have multiple sgRNAs, and you'll get sort of convoluted effects. And then you can use antibiotic resistance or sorting or any other method for enriching for infected population. And so that gets you to this, where everything is either sort of a negative control cell or a cell containing some targeting perturbation. So just wanted to go through a few very typical screen designs and the things that we think about when we're planning to do a screen. So the simplest possible one is a growth screen. You have your population, and if you just had two sgRNAs in your pool, you could have one be a non-targeting guide and then one targeting like a ribosomal gene. that's essential for cell viability. And then it just as simple as letting them grow over time, over a number of doublings, and your negative control cells will double normally because they don't have any genetic perturbation. But if you don't have a ribosome, you'll probably either die or just be static in culture, and the negative control cells will outcompete everything. And then you can sample both of these populations and read out the distributions there. And so one important concept throughout all of this is coverage. So if you have one sgRNA per cell, you also don't want just one sgRNA to be, if that's one experiment, you don't want to do that once, because if that cell dies, if you leave it stuck to the dish, then so much for testing that gene. So you really want what we call library coverage. So you're not just growing the cells in a cartoon, they keep dividing and you have to keep splitting them back. So your cell number varies over the course of an experiment. And you never want to have just one sgRNA represented once. We really go for more like 500 to 1,000. So that's at the minimum point, at the sort of statistical bottleneck, which you might introduce every day or every other day if you're splitting the cells. You never really want to go low. You need to have a lot of cells to have good statistical representation. And that's really the main challenge with all of this is while it scales much better than a lot of other technologies, you still need to have a lot of cells growing. And I'll do some math in a little bit to estimate how big an experiment can get. The next kind of screen is very similar, and Jonathan had a lot of nice examples of really how well this works. Are drug or toxin selection screens. And there you start out with the same starting population, but this time you can treat either with vehicle, which ideally shouldn't do anything. You might see some loss of these essential genes, but the vehicle itself won't have any effect. Or you treat with drug, and your population will evolve to one that is dominated by genes that, when knocked down, are resistant to your drug of interest, or when knocked down, have a synthetic lethality relationship with that drug that really synergistically die when targeted with the drug. And actually, I introduced the concept of coverage in the last slide, but even here it's even more important, because every time you hit with a drug, you're killing cells. Cells respond stochastically to the drug at some level, and so you have a really strong bottleneck where you can kill a lot of things randomly and start to lose the true effects that you're trying to see. So minimum coverage is key. Jonathan alluded to the different sorts of dosing schemes that you can think about. LD50 is conceptually very nice, so you're at the middle of your sensitivity curve, and if you hit a drug at the LD50 and then change the sensitivity very slightly, you change how the cell responds quite a bit. You can get complete resistance or complete sensitivity with just very minor changes. And of course, it makes it easier because you're killing the cells less. But a lot of cancer biologists especially say, well, I'd never use an LD50 in the clinic. That would kill my patients because I'm only getting rid of half their tumor. Can I go harder, really get most of the cells and only see what grows out as a small fraction? And it's definitely possible, as approaching an LD90. But then if you imagine the coverage being an issue, you need to start with 10,000 cells in order to have 10% be 1,000 cells. So these swings that happen here become much, much more dramatic when you're not just splitting a defined amount, you're hitting them with a hammer over the head in order to see what promotes resistance. And then one other nice mode of screening, and I know Jacob assigned this paper for his talk, so I won't get into it much. But FACS-based screening is a really nice approach for looking at more specific biology. So you have a population pre-sorted, you have your sort of background distribution, and then you can compare it to running things on a fax, sorting, a bin as you might often do with trying to isolate single cells or a polyclonal population, and then just sequence that population. And so here, these are sgRNAs targeting CD13, a cell surface marker. You just add a stain to see CD13 on the cell surface, and if it disappears and you sort that population, things that have destroyed CD13, like sgRNAs cleaving in the middle of the transcript, will become really, really strongly enriched. And there are a lot more interesting biological applications for that, reporters and that sort of thing. So you can start getting into much more specific biology with a FACS-based screen. So again, the math is really where you have to be careful in the design, because so first you have to decide how many genes you want to screen. And so let's say we're doing a genome scale screen, 20,000 or so. Then you don't want to do just one reagent hitting that gene, one shRNA. If you have lots of off-target effects, you have no way of knowing if that sh is getting the phenotype from the gene or from an off-target. And the same principle applies with CRISPR. We know it much less off-targets, but you need independent experiments to be able to really say this gene is causing a phenotype. And then, of course, the minimum coverage issue, so number of cells representing each sgRNA. So if you have 20,000, if you have a pretty typical number, 10 sgRNAs per gene, in order to get really a robust statistical readout on whether that gene is a hit or not, and then a typical coverage number like 1,000 cells per sgRNA, that already puts you at 200 million cells, which is a lot, but it isn't too bad. And there are lots of ways to culture that in the lab with our K562 cells, which are in suspension. We actually grow them really easily in spinner flasks that all you need is like 400 liters of culture in a spinner, and you grow it for a little while, and you do the sequencing, and it's really very easy. With adherent cells, it can get a bit more tricky because you probably need multiple plates in order to grow 400 million cells. And I know it says 200 million here, but you need 400, and then you split back to 200. Would be the issue. But of course, again, to really do a good scientific experiment, you normally like controls, and you normally like replicates, and you probably need conditions of treated and untreated and all of that. So it does start to add up. It's an incubator full of spinner flasks or an incubator full of stacks of plates. But I think, oh yeah, sorry. Yeah, no.
I was wondering, have you considered using microcarriers?
Yeah, and some collaborators have used that pretty successfully.
We were thinking about using that, but then I didn't know if they had an issue with absorbing, say, the guide.
I think the best protocol people have used with this is sort of infect the population, seed them very sparsely on the microcarriers, and then they actually can grow out, because there's so much surface area in one of those in microcarriers, you can grow out without splitting at all over the course of a screen, and then at the end just harvest the genomic DNA right off the beads. We can talk about this more, yeah, but it's definitely a practical option. Yeah.
It wouldn't save you any cell culture work, but have you ever thought about just chopping up the pool into smaller subpools just to give you a little more flexibility?
Absolutely, yeah. So a lot of the libraries that get published and are available on AdG now are split into subpools. usually based on some biological categorization, and it's often hard to get the perfect division. You want sort of non-overlapping pools, but at the same time, how do you divide up the genome in a rational way? So that there's also the final option of, picking your own set of pools and ordering it, and you have a defined genome scale or subgenome scale library that you want to screen against. So there are definitely ways to do that to make it easier. I think if you can, it makes your life much easier just to, get it over with. So as the final step, and probably one of the more daunting for labs that don't do a lot of sequencing is how do you actually read out these population distributions from a frozen sample of cells that you've taken at the beginning of an experiment and after hitting with drug. And so you have your complex population, and easily you can harvest genomic DNA. But what I think often gets lost is that you don't have to sequence everything. You have a very defined set of transgenes integrated into the genomic DNA, and you know everything about them because you designed them and you put them into lentiviruses and you put them into your genome. So really, there are lots of protocols out there. The basic ideas is that you just do a PCR off the genomic DNA and get your complex pool just as these barcode fragments with the Illumina adapters or whatever sequencing technology you're using. And then it's just a matter of sending to a sequencing facility or a core. It is a very short, usually 20 base pair barcode, so it is kind of the cheapest option in screening is lots and lots of very short reads. And the coverage concept still applies here. And then you just align back to the library and just count how many cells here gave you the sgRNAs here and in each condition. So you start with your untreated, and then you see, these doubled in count number that are promoting resistance on some level, whereas these negative controls really didn't change at all. Yeah.
When you count, do you count exact matches only, or do you allow for sequence matches?
Usually exact matches only. Yeah. The nice thing is, if those mismatches were present in your library, then you can just throw them out because they're, yeah. So the question is which technology to use, and obviously that isn't necessarily the right answer at this workshop to say RNAi. It is a really nice technology, and I will say it's still quite useful in a lot of contexts, but it does use the endogenous machinery, which as Jonathan mentioned, can be problematic in a few ways. And of course, off targets, variable knockdown, all the usual things that people who've used RNAi a lot like to bring up. So of course, the next option is Cas9 wild type, active, creating double-stranded breaks, and then really what actually makes the mutation is the repair pathway, which is randomly creating indels in your gene of interest, and is ideally total loss of function. But I'll get to the asterisk on that. And then the next option would be CRISPR-I. You repress your gene of interest in a very targeted manner, and you can do so for all alleles kind of evenly, and I'll get into that too, and then CRISPR-A. And so I think Luke yesterday talked really about the basis of these technologies and some sort of individual benchmarkings that we've done on all of these to really characterize how they're working, what the off-target rates are, and what we like to do with them. But in a screening context, there are a few more other things to think about. They all produced lots of papers. and a nice review, and actually a couple of reviews. This one from Feng Zhang in Nature Review Genetics of this year, which I encourage you to look at and sort of see the really a nice view of all these different screening platforms in detail. So one quick way to differentiate all of these is how many components there are. So shRNA is fantastic because it's really just one lentivirus. You stick it in the cells, it co-opts the endogenous machinery to process, to target. So if you don't have a lot of wiggle room with the cell line, it's a primary cell and you really can only infect it once, shRNA is still fantastic. And it continues to be improved. And if the one limitation with improving shRNA is the more effective you make the knockdown, usually the more effective you also make the off-target knockdowns. So there is a ceiling on that, but as a one-component system, it's kind of really elegant. CRISPR and CRISPR-I are, of course, two-component systems. You have your Cas9 and your sgRNA, and so you need to do a little bit more engineering with the cell line to really get both and make sure that both are working together properly in the screening context. And again, if you have one of them inactive, then it really is going to dampen any kind of readout that you want to get because these are just going to be cells growing in culture that aren't listening to whatever sgRNA perturbation you want to put in them. And then finally, CRISPR-A at single copy, both our solution to CRISPR-A and the Zhang lab had an alternate solution. Both of these are three-component systems. Just having one Cas9 targeted to a locus alone usually gets you at most 2- to 3-fold activation, which, depending on your gene of interest, might be enough, but isn't necessarily a robust way to say, I'm overexpressing this gene quite a bit. So in order to amplify the signal, there are two different approaches, and I'm fairly sure Luke talked a bit more about the details. I'll just say it's a three-component system. It takes a lot of work to get a cell line working, but then you can activate nicely. And of course, the orthogonality goes in favor of CRISPR because you're not co-opting the cellular system, so you don't have any sort of toxicity by overloading the RNAi pathway or anything like that. And then the real thing to think about when doing a screen is what is actually happening to each of the cells that you're targeting with a particular SH or sgRNA. And so with this being a representative FACS plot of if you're targeting this protein, this is the wild type expression distribution. This is a complete null expression distribution. And when you add an shRNA, you're knocking it down. So you're shifting your wild type distribution down, not true null, but everything is sort of dampened quite a bit, and that can be enough to get a phenotype. With CRISPR, you're starting with, again, 2 alleles of your target or more in lots of cancer cell lines. You have triploidy and aneuploidy, so you have multiple alleles to hit. So if you are making random deletions, sometimes they'll be out of frame and pretty much destroy the protein with a nonsense mutation. So that would be a true null, and that is an attractive thing, even if it's not your whole population. Even if you're getting heterozygotes, it's attractive to have a true null, especially if you have something where you're looking for resistance, where a true null is the only thing that will give you resistance, and you need to see it grow out of the population. You won't care about these as much because you have a knockout and it'll proliferate and you'll get really great signal because it's completely resistant to a drug or gives you a completely clean signal on a FACS assay. But it is very complicated if you want to interpret, is this gene essential or not? Because these cells will die because they're completely knocked out for an essential gene. But a heterozygote, or even a gene that basically hasn't been perturbed at all in some fraction of cells, might dominate your population, and you're looking for cells dying, and these aren't going to die because they're kind of a mixed bag of random mutations which aren't always deleterious. So that's sort of the thing to weigh with an experiment where you're making a random mutation. With CRISPR-I, it's basically the same as RNAi. We just drew the histogram a little bit further left. Your readout is going to be similar to RNAi, but hopefully much more potent repression and much better off-target profile. So you have a much better signal-to-noise readout when you're actually looking at your screen. And then, of course, CRISPR-A is going the other direction and is also sort of a uniform modulation of expression, either turning it on, turning it off, rather than this sort of mixed bag of alleles. So there are lots of review papers about this now. There's one nice one from the Pelkman's lab that is one of my favorites. And it really gets into all the various things that you can weigh when designing an experiment. And of course, the Zhang lab paper not only gets into this in nice detail, but also really highlights now there's a proliferation of libraries. You go on AddGene, and there's a very, very long page of libraries that if you're an academic lab, you can buy and introduce into your cells and basically have something that's already made for, in most cases, most of the genome. Some libraries are smaller and some libraries are full genome, but you can order the sublibraries, and then there's always variation in sort of these other parameters that I've talked about earlier in terms of the math of how much you're targeting a gene and how much cells you have to culture and all of that. So I think that wraps up the first morning part of the talk. So pooled screens are a really neat concept, sort of an elegant way to be systematic, but also only have to deal with one experiment, one pot. And the great thing about that is you don't need any sort of specialized technology. It's mostly molecular biology and tissue culture, and then you have to send it for sequencing or have a core facility do the sequencing. But it really, except for the cost, is kind of a universally usable technology. And just the main highlights to think about is what technology is best for the phenotypes you're trying to see, making a really good cell line in order to do the screen, and then making sure that you have sufficient cell coverage to actually be able to do statistics and have good readouts at the end. So with that, I'll jump in a little bit to a few of the papers that introduced and characterized these systems, and then spend the rest of the time on some new exciting things that are emerging with these technologies, and also other applications that sort of take the same concept but turn it on its head in a few different ways. So the first paper that I actually listed on the lecture syllabus guide is this paper from the Lander and Sabatini labs, where they introduced CRISPR-Cas9 active nuclease screening for the first time in tandem with the Zhang lab paper. But both papers cover somewhat similar topics, so I'll just focus on this one. So one important thing, as I was saying, is indel introduction is pretty stochastic. So they nicely put that out in front and actually sequence some loci and try to look at how frequently you get in-frame, out-of-frame deletions. I think this picture is a bit rosy compared to what other labs have seen. So they're seeing a 3 base pair deletion is incredibly rare, which would be an in-frame deletion that would sort of not affect your gene at all, but would be an underlying confounder in your assay. But
Max, can I just
yeah.
say one thing? In-frame deletions in a critical part of a protein coding sequence are going to be nulls too.
Yeah, get to that.
Okay.
Yeah. Yeah, no, there's been good work using that fact to actually to look at proteins on a single protein domain level. But if you're not designing your library around domains, you'll hit a lot of areas where one amino acid might not be too much of a hit. And they also sort of go into, in this paper, some very early library design parameters, and just as a demonstration, that when you're designing a active Cas9 library, you're focusing, of course, on coding exons, trying to hit exons that are present in every isoform of the gene that you're targeting, and just employing some simple metrics here to generate this first initial library. And it really worked pretty nicely, especially for this positive selection screen. So they took their library that they synthesized, introduced it into 2 cell lines expressing Cas9. This one, KBM7, is haploid, so it makes their lives much easier in the indel distribution because you only have one allele that you have to target. And then they also targeted a diploid cancer cell line. And in just a screen for a topoisomerase inhibitor, and a topoisomerase inhibitor that actually is toxic if it binds active topoisomerase, knocking out the target was strongly enriched. Those cells were very resistant. And this is kind of almost the raw data view of the output of a screen like this. So it's just the counts of the sgRNA in an untreated population against the counts in a treated population. And so if it's treated with the drug and it's resistant compared to the background distribution, you get really nice enrichment. And over several distant, each dot is an sgRNA, and if you are targeting a gene with multiple, you'll get hopefully the full set shifting up compared to the rest of the cells. And they see that as well with the diploid cell line, although you could argue that it's a little bit more of a damped signal, but it still definitely complete separation, especially these green dots are a few negative controls that they're really totally separated from. They also did a good job going in and looking and making sure that they could see negative selection. So that's really where you would expect indels to, without effects being a problem, and where you're knocking out essential genes, so that DNA replication, RNA polymerase, all these essential components do drop out. This is just a gene set enrichment analysis plot showing that on the depleted end, almost all components are depleted relative to the general library. So even with the sort of caveats and facts plots that I mentioned, you can really do some nice loss of function assays, especially in a haploid cell line. So are there any questions on sort of the active Cas9 screening? All right, there have been quite a few of these examples, and they do varying levels of characterizing their system and showing nice examples of genes that make a lot of sense, either that are known or that can be validated, that are true hits that have been discovered, and both in human and mouse have been now done pretty extensively with active Cas9. Switch gears and talk for a little while about our CRISPR-I and A screening platforms. So we really didn't know. We had this nice technology. We knew we could, in some cases, knock down a gene well, but we didn't really know what the parameters of our system were. Where to design an sgRNA to actually be able to have an effect on a gene, and to be able to do so kind of without knowing anything about the gene except for the annotation. So we started out with 2 different types of libraries. First, we used a small library targeting a small number of genes with a large number of sgRNAs, basically every possible one that we could conceive of targeting that gene, and did sort of a tiling assay. And so I'll get into that a little bit later in terms of what I mean by tiling assay, but just generally, you can look at every possible parameter in your system if you use every possible guide. And 49 genes may or may not be representative, but it provided a really nice guide that for CRISPR-I, for instance, right around a TSS is where you have all of the activity. So this is sort of a random distribution upstream of the TSS, and then within 1 kb or so is where all of our activity was present, and this is average of our 50 genes. And so it made it very clear how to design a library. We also did some experiments looking at specificity, and then another aspect to designing a library, which I think Jacob will get into likely more, is off-target scoring and how to design a guide that is very specific, both highly effective against your gene and unlikely to be effective against any other locus. So this was just a sort of systematic mismatching experiment that we did also in the same pool in order to see what bases, when mismatched, affected guide efficacy the most. And you can use that to predict against the rest of the genome. So we were able to use this to simulate what a library would look like on these 50 genes, and saw that it would almost invariably be superior to the same genes with shRNA that we had previously seen in screens using the same toxin ricin. And so all of that fed into our genome scale libraries, where we took 16,000 genes now, and instead of hitting every possible space, we used these rules to just pick 10 guides. And unfortunately, we found with these rules that actually INA, interference and activation, somewhat makes sense, have to be in different places. If you're blocking polymerase, you can't really activate it all that well, even if you have a strong activator, you're blocking polymerase. So INA sort of occupied non-overlapping spaces near the TSS, and we've made multiple libraries, one for each. And so what do these libraries look like? So this is similar to the Lander paper, looking at just growth of cells when you've knocked down knocked down every gene, but what genes are essential, what genes deplete when you knock them down. And so these are just replicates with each sgRNA being a dot here. And so you can see we get for a large proportion of sgRNAs, we get very well correlated and strong knockdown away from this distribution of negative controls. And of course, so I guess a few important things. One is that our targeting was very reproducible. Obviously, you don't want a system that doesn't correlate with itself, experiment to experiment. But the other important thing was that the negative controls were markedly uncorrelated. And so this is not something that was often seen with shRNA, because even if you've designed an shRNA that doesn't target anything, it still has off targets to other genes. And so you'd often see, actually, even though they were usually centered around 0, they would correlate with each other, which meant that there was some true signal in the negative control. And so that suggests to us that we were being very specific in both our targeting guides and our negative controls. You could, of course, argue that we designed these specifically to be bad, because if you made a really, really bad negative control that would never hit anything, you would get a random distribution. So we also highlighted a few different sets that we didn't expect to be essential. These are female cells, K562, so no Y chromosome, and indeed they didn't correlate with each other as well, and they were all around 0. And then also olfactory genes seemed unlikely to be hits. And then aping the Lander paper, we did these gene set enrichment plots and similarly saw the expected gene sets depleting. So sgRNAs targeting ribosome, replication, all of those kind of came out as a set as strongly depleted away from the general population. Yeah.
When you're designing your guide RNAs around the transcription start site, do you, because of the reversible nature of activation and inhibition, can you relax the criteria? You're not as worried about an inhibitor binding some anywhere outside of that transcription start site, or do you use the same stringency for the nuclease designs?
In terms of off-target prediction?
Yeah.
So originally was using sort of the same stringency, like looking against the full genome, making sure it wasn't binding anywhere. It started getting restrictive, and so I in newer library designs have gone to sort of a hierarchy of stringent for any big sequences near a transcription start site, and then somewhat more relaxed for the genome. You do have to be careful if you're very relaxed. There are a lot of sign and line elements, repetitive elements in the human genome that if you don't filter out at all, you'll bind everywhere and probably not be very effective. So there always needs to be some filtering. Then we did the same exact growth screen using CRISPR-A. And so now each dot is a gene level view, so averaging the most effective sgRNAs for CRISPR-I and CRISPR-A. And so I is on the x-axis, and this is the same sort of depletion ribosome DNA replication that we expected. And we can see immediately that what kills cells when overexpressed is completely orthogonal. You could argue that you might expect them to be anti-correlated, something that you turn up or turn down should give, sort of, if you're turning these down and it kills the cell, if you're instead turning it up, it should make the cell grow faster. That in cancer cells usually isn't the case. It's very hard to find anything that'll make the cell grow faster because it's sort of optimized for cell growth. So we don't see any of those, but what we do see is a very nice set of a smaller set of genes that are very specific for the biology of the cells that we're looking at. And so we find classes of developmental transcription factors, tumor suppressors that, when overexpressed, cause the cells to stop growing, and also some centrosomal genes, which make for very nice pictures. So this is an activation of polo kinase 4, not in K562s, in a retinal cell line that Jonathan showed some microscopy from before as well. And so it's thought to be involved in centriolar duplication, and so activating it causes some crazy mitotic figures with lots and lots of centrioles. So just in summary, the inhibition and activation give some really nice complementary different sets of information that can tell you both about essential genes, and then we've done lots of drug and toxin screens to further build out both biology that we're interested in and sort of what makes a good screen and a good library. And one final point on CRISPR-I is that it's nicely inducible. So it have the Cas9 under DOX induction. When it's not induced, and you're targeting, these are essential genes again, so if you're not inducing Cas9, you get no depletion. But if you add DOX and start knocking down essential genes, you very rapidly lose these cells from the population. And so it's very nice if you have a system where you have some temporal control. You can get all your components into the cells and then induce and see what happens right as you've induced knockdown. And that's actually also reversible, at least to some extent. Taking off docks can restore full expression or near full expression. And we can also use this in screens. This is just a small essential screen, so the gray is negative controls and black are targeting guides. And so upon docks induction, all of a sudden, same deal as this, except in a pooled screen. You really see complete depletion of sgRNAs targeting essential genes. And as I mentioned earlier, there are now multiple ways to do CRISPR-A. So our system relies on intracellular antibodies to make sort of a super complex of VP64, recruit 10 VP64 activator domains. The Zhang lab came up with this nice system that's also 3 components, but has one VP64 and then a few different domains to make it, the signal amplification happens through the fact that you have some heterogeneous signals being recruited rather than really hitting it over the head with VP64. But so that's sort of a rundown of the various technologies that have been fed into these CRISPR screens, and happy to talk more about them. But I thought it might be nice to, and as one final point, it's kind of continuously evolving field. There's going to be probably new libraries every week for the next few years. We're definitely in the progress of improving our library designs, and I know several sort of nuclease screening groups are interested in that too. So, there's always room for improvement at this point. But the technologies are starting to get to a pretty, I don't know if mature is the right word, but robust, easy to use setup. So yeah, how about what else we can do with these screens? What other ways can we make them more informative than simply a list of what's killing a cell, what's causing resistance to a drug, and what's going to be kind of coming out in the next few years. So talk about these first 3 points in a bit more detail. So expanding beyond the protein coding genome, some in vivo screens that have been coming out and that will probably get improved in the next few years. Higher content screening, so rather than just one phenotype, can we in a pooled fashion ideally extract more information out of a genetic perturbation? And then finally, I think I'm not going to talk more than just the bullet point, but as Jonathan mentioned, genetic interactions are a really, really valuable tool and have been done a lot in yeast. And now that we have all these nice, robust technologies and lots of options to do screening in human cells, it is for sure going to be a transformative tool to be able to find new functional associations using genetic interaction mapping in human cells. And it'll be interesting to see also with new non-strep pyogenes Cas9s, what various tricks people can do building on the repertoire of what already exists. So in terms of expanding beyond the protein coding genome, there are sort of two classes of non-coding genes that I'll mention. One is microRNAs. So they can be pretty hard to target. Maybe sponges are somewhat effective, but in general, there haven't been any good systematic tools to probe microRNA function. So the Gecko libraries actually now include sgRNAs targeting about 1,800 microRNAs, and I'd say that actually this is a really nice tool for probing them because microRNAs are very short, 20 to 40 bases of specificity, and really they usually have a pretty small seed region that actually determines all of its effect. And so if you can mess with its specificity in just even subtle ways, it should completely or almost completely disable microRNA function. And it's come up as hits in a few screens that they've done with this. I don't know if they've followed up more in terms of, actually is the microRNA that they're hitting the relevant hit, but it seems like a really nice approach. And CRISPR-I has been shown also to be able to repress microRNAs, but not in the same directed fashion. So CRISPR-I, you can only really inhibit transcription and do so from a transcription start site. Lots of microRNAs are either processed from introns or processed from host genes that have multiple microRNAs. The sort of most famous example is the Mirror 1792 cluster, which has a lot of implications in immunology and in cancer, but you can't, with this, tease apart which microRNA is doing what, but you can perturb the locus, at least broadly, knocking down all transcription from that cluster. And then the second class of non-coding genes are lincRNAs, long non-coding genes. And these have been really difficult to study because there are not that many tools that can perturb them well. So shRNA is mostly effective against cytoplasmic transcripts because it's designed against mRNAs, and so it's variably effective. And so we've found that CRISPR-I is actually sort of a uniquely suited tool for perturbing lincRNAs. And so just to start out, we predicted a few guides just against a handful of well-studied lincRNAs just to see if we could actually perturb their expression. And in some cases, we can get really, really strong knockdown just right off the bat, testing to see if you can knock down something like exist, which normally is expressed on the X chromosome and spreads along the X chromosome. And if you do FISH, that's normally what we see, that each cell has sort of a focus of exist expression, but knocking it down makes it completely non-visible, at least on FISH, and by qPCR is almost totally gone. So that's going to be sort of an exciting set of tools to be able to figure out what long non-coding RNAs are actually doing and which are required for cellular processes, and both screening and I think as an individual tool. CRISPR-A should work in principle, and I think some people are working on that. We haven't tested that one way or another in our hands, but if CRISPR-I is working, and I don't see why you couldn't activate a lincRNA too and see what it's doing. And then finally, nuclease is going to be tricky because there's no coding frame to be able to do frame shift. So there's no guarantee that a small indel is going to disable a lincRNA. But you could imagine doing paired nickase, cutting out pieces of long non-coding RNAs, cutting out exons, and trying to scale that up to sort of the number of lincRNAs that have been described and try to get at function that way. Another exciting field is in vivo screening. So this is actually an example from RNAi from Elaine Fuchs's lab that I thought was really nice, where they actually transduce mouse embryos with the full genome scale shRNA libraries, grow them for 9 days, and then harvest the epidermis, being the Fuchs lab, and look at what cells are both required for normal growth, and they also add oncogenic RAS and see what genes are responsible for oncogenic growth in utero in vivo. And so this is sort of a really, really cool first attempt at a whole organism shRNA screen. For sure, the assumptions that you make going from a pool where each cell should be acting independently to an organism where cells next to each other are talking to each other and might be expanding clonally and might not, really changes how you think about what's happening in your screen as it goes along. But they see kind of the same ribosomal genes, spliceosomal genes dropping out as you would expect from a pool. So it holds a lot of promise for being able to look at homeostasis in an organism. Recently, the Sharp and Zhang labs have published first a mouse expressing transgenic Cas9, and then an in vivo screen, not using that transgenic mouse, just introducing lung cancer cells that have been transduced with the library, and then looking at distant metastases from the xenograft. And it was definitely a cool idea, and sort of funny that they're injecting lung cells into a hip and then looking for it to metastasize to the lung. But I guess lung cells like being in the lung. It's not the typical route for metastasis of lung cancer cells. But they get both some nice hits and some head-scratching hits that I think make it look like this sort of screening isn't quite there yet. It's hard to see. One of these is an olfactory receptor, which we've used normally as a control for this sort of thing, and maybe has other functions as well. And then another one is a core polymerase component, which should really have killed the cells, not encouraged metastasis. But they didn't go into validation of those hits, so I don't know if maybe these are true effects. But either way, this is a nice approach, still kind of noisy and needs to be sort of carefully characterized going forward. And then finally, one nice, somewhat in vivo or ex vivo approach that really just came out a week or two ago is from the Regev lab, where they are interested in dendritic cell pathways. And so they take now the Cas9 transgenic mouse, harvest primary cells from them, introduce the library, and then do several stimulations, sorting, kind of typical immunological techniques to be able to separate different populations. And they do, the reason I add this paper is they do a really nice job showing sort of how comprehensive it's perturbing these pathways. Almost every gene that should be showing up is showing up, and they kind of carefully go through and validate. And so if you're ever sort of curious how to validate a screen, this is one nice example of carefully doing that. The next future direction of screening is high content screening, and I've talked to a few people in my small group who are kind of experts in that, and definitely don't know too much in that field, but I think there are opportunities there to be able to either do arrayed high content screens or possibly some pooled approaches that let you scale up and pick out more than just whether a gene is causing life or death. So this is also a review about high content screening, and one question is, can single cell screening, where you get a full expression profile from a single cell sequencing, be done as a pooled screen? And I think this will be a relatively easy step in adding to the content of screens. The throughput might be low because it's hard to do single cell sequencing on 1 million cells, but it is an easy concept. If you're already sequencing the whole cell, if you can put the barcode in a place that it'll also be read by single cell sequencing, you immediately have both your genotype and a full expression profile, and you can think of sort of a host of things that you might be interested in it directly tying perturbation to RNA-seq. The other one that I'll mention is microscopy. You can get thousands and millions of parameters just by taking a picture and figuring out cell size and what its neighbors are and what subcellular structures are looking like. And this is for sure going to be hard to actually pool, because you want to know what the cell you're looking at looks like and its genotype, and it's sort of hard to go from a microscope to a molecular barcode. But there are a few ways that I think people will be able to brainstorm and troubleshoot through, where you could microdissect a gene that has a high content phenotype of interest, like a damaged subcellular organelle, and pick it out and then sequence a barcode and see what things are coming up that are showing a phenotype of interest. All right, so for the final 15 minutes, I'll kind of get into some other concepts of pooled screening, where you're using the advantage that you're in a pool, but you're not necessarily caring about hitting every single gene. You want to drill down and get some more high resolution information. So I alluded to earlier with with tiling screens, if you can really look at every single guide that could possibly exist along a gene body or along a TSS, you can both figure out what is making your system work and use this as a really powerful benchmark for any machine learning, because if you have complete information but you hide that from your machine learning, and stop me if I'm kind of babbling about machine learning, but if you have perfect information and then you try to predict what good guides are going to be in a particular locus, and then you check back and see, I picked these top ones, then you're doing pretty well. But if you're kind of doing a mediocre job, you'll see immediately, that there are a lot more effective things that you should be finding that you're somehow missing, in your bioinformatic approach. And so, this is one example gene and an example of the metagene analysis that can really be informative for characterizing and designing a system. And of course, one nice thing about it is if you're not really designing it for one particular technology, you can really, do a tiling screen, the same screen with every single different activator, inhibitor, different Cas9s potentially, active Cas9, and really sort of saturate both the locus and the information that you're getting. So this is the same exact library, same exact gene, but with the activator, and now you see both different sgRNAs are working, and the phenotype is exactly the flip, because you're activating rather inhibiting, and in this context, getting the opposite phenotype. This has also been done effectively with nuclease. I'll leave this for Jacob, but this is a nice example of tiling as well, in David Root's FACS paper. And then the other sort of information, a little bit less dry than actually, what guide is working well, is defining functional elements. So there are some reports. I think there needs to be a lot more on actually, if you're tiling with dead Cas9, you could imagine figuring out where regulatory elements are. If you block a particular regulatory element and see a gene expression change or a phenotypic change, you could really map out enhancers in a particular region. But that's pretty far off. One nicer example of this is tiling a transcript with nuclease to find functional domains, and this is back where what Dana was mentioning. So let's say you have on average 30% of your mutations from active Cas9 get repaired in frame, and that seems to be what they've seen at the locuses they've tested. This is in a Nature Biotech paper from the Vacok lab. And so if you have 30% in-frame indels, you have 2 alleles per cell, normal diploid cell, then you sort of square the chances of not getting an out-of-frame indel, and actually 51% of your cells will have at least one in-frame indel. And so in a lot of regions, this might not be enough to actually compromise protein function, but maybe in some regions where each amino acid is critical, at least to be present structurally for the enzyme to have a function, you might be able to see a different, more pronounced effect. And so that's exactly what they've seen. So they tiled a number of example genes. This is a chromatin modifier, both chromatin modifiers really, and where they highlight the bromo domain, the relevant domain in bromo domain 4, they see much more depletion. So it's an essential gene, so they're looking for just depletion from the population of the sgRNA. And while you see some depletion throughout the transcript, really compromising function even with an in-frame indel is much more pronounced if you are in a functional domain. And so you could imagine if you didn't have this here, you could do a pretty good job of guessing what areas of the protein are interesting, what you might want to do some mechanistic studies on. And the nicely show that this is because so for this sgRNA not in a functional domain, the in-frame deletions don't deplete when you sequence actually the indel frequency, and the frame shift mutations do deplete. But at the functional domain, they track exactly with each other. You get the in-frame indels and the frame shifts completely drop out. And you could say, why don't people design libraries around domains? And really, you can start thinking that should be the case. Not every gene has their domains nicely mapped, and sometimes you worry about what if you have an in-frame indel in your bromo domain, but it still has a scaffolding function. You're still have something active in your population, even if it's not the enzymatic function. And then finally, one really nice paper from Jay Shunder's lab at University of Washington kind of took the library design on its head, and instead of saying we want all these sgRNAs making mutations everywhere, what if we really saturate one region, make the library not of the sgRNA, but of a homologous recombination template? And so that's what they did. They added CRISPR just targeting one locus, again, in an essential gene in this case, and said, what if we introduce random hexamers in an HDR library? So homologous repair will, even if it's not perfectly efficient, will introduce this hexamer into that particular locus, and then you can sequence and see, at the beginning and the end of an experiment, what happens to the specific DNA sequences at that locus. And so this has been sort of a major goal in cancer biology, because patients always present with things like a V600E mutation, but why? Why V to E? Why right there? Why not a neighboring amino acid turning to an E? So saturation mutagenesis is really kind of neat way to ask that question for systematically every amino acid substitution in a particular area. So sort of data that they get is like this. So their library is designed with some synonymous mutations, some missense mutations, and nonsense ones. And if you introduce them into an essential gene and in a haploid cell line, just to make their lives easier, you start enriching for both synonymous and missense mutations, and you get almost completely no nonsense mutations at the end of growing this population. And it's pretty cool, this base, this boxed in green, is normally a glutamate, but it's completely fine if you put any codon of aspartate in there. But everything else, except for this guy maybe, is non-functional and drops out because you've killed the gene, killed the enzymatic function. So this will, sort of another way to say I only care about this one gene, and I want to, know everything about it. Could tile the whole transcript and find where the relevant domains are, and then you could drill in and say, I want to know what every mutation at this spot is doing. So with that, just again say, multiplexing CRISPRs is a really easy way to get a lot of information and to enable discovery. And we talked about some emerging technologies and some other uses, and take any questions.